In talking about cancer and genetics, it's helpful to know what pre-existing perceptions people have about their genes and cancer. Tell me a little bit about what you know about genes and how they relate to cancer.
Well, I know it could run through families. My dad and my aunt had cancer, so I will probably get cancer too.
Perhaps not necessarily. Not all cancers run through families. In fact, 90% of cancers are sporadic or out of the blue. These cancers typically arise later in life due to the wear and tear of the natural aging process. However, in some families, there are genes that can increase the risk of cancer. When we see many members of a family with the same kind of cancer, especially if they are all on the same side, or if we see young diagnoses of cancer, we start to suspect a hereditary cause. This hereditary form of cancer makes up the remaining 10% of cancer cases. So, if your aunt is your father's sister, and if they both had related cancers diagnosed under 50, we have a strong foundation upon which to recommend genetic testing.
How do people even get cancer?
Our bodies have natural defenses to protect us against cancer forming. They are known as housekeeper genes, or DNA mismatch repair genes. These genes form a protein that helps correct mistakes in our DNA during the replication process. In hereditary cancer, individuals are born with a mistake or mutation already in one of these housekeeper genes, making it less likely to do its job and fix mutations. As your cells proliferate, the mutations accumulate, and cancer is often the result. Let's look at an example of a hereditary cancer syndrome called Lynch syndrome to get a better idea about how genes relate to cancer. Lynch syndrome is an autosomal dominant genetic condition that has a high risk of colon and rectal cancer, as well as other cancers such as endometrial, ovary, stomach, small intestine, brain, and skin cancers. It is characterized by germline mutations in the MLH1, MSH2, MSH6, and PMS2 MMR genes. In the United States, about 150,000 new cases of colorectal cancer are diagnosed each year. Approximately 3 to 5% of these cancers are caused by Lynch syndrome. In individuals with Lynch syndrome, their lifetime risk of colorectal cancer is up to 80%, stomach cancer between 11 and 19%, with other Lynch cancers between 1 and 7%. In women, the risk for developing endometrial cancer is between 20 and 60%, and ovarian cancer between 9 and 12%. A diagnosis of Lynch syndrome does not guarantee cancer development, but it does pose a higher risk for cancer when compared to the general population. Let's explore the ins and outs of Lynch syndrome by walking through a research article published in April 2016. But before we start, it may be helpful to discuss how Lynch syndrome is diagnosed. Lynch syndrome can be diagnosed in one of two ways. The first way is simply a genetic test. An individual provides a blood or saliva sample, which is then analyzed to provide a genotype at MMR loci. Another way to diagnose Lynch syndrome is to analyze MMR protein expression using immunohistochemistry staining, or IHC. If individuals lack protein expression, a diagnosis of Lynch syndrome is assumed.
So if someone shows presence of the protein, they don't have Lynch syndrome, right?
That's a very good question, Sal. The answer is we can't know for sure. Let's think about why. Why would lack of protein expression be sufficient to diagnose Lynch syndrome, but presence of protein is not? Well, perhaps someone in our biochemistry class can discuss this question in the forums. So, returning to our study, researchers in Mexico took blood and tumor samples from their patients suspected of having Lynch syndrome due to strong family history of cancer. IHC was performed as a screening process to select those that most certainly had Lynch. Of the pre-screened patients, 3 showed loss of staining for MLH1 and MSH2 in tumor cells. Patient LS23 and LS41 showed absence of nuclear staining for MLH1. Patient LS52 showed absence of nuclear staining for MSH2. Once the patient sample was established, the researchers' next goal was to sequence their DNA to determine what mutations the patients carried. Genomic DNA was extracted, and 19 exons of MLH1 and 16 exons of MSH2 were amplified using PCR. The fragments were then visualized by gel electrophoresis. An aliquot of the PCR product was then placed into a DNA separation column in a process called DHPLC, or denaturing high-performance liquid chromatography. The chromatograms of each fragment were compared to the wild type. The chromatograms that contain heteroduplex peaks and shorter retention times when compared to the wild type underwent Sanger sequencing to characterize the mutations. The PCR products were purified and then assessed using gel filtration. The purified products were sequenced and subsequently analyzed using a genome browser hosted by the University of California in Santa Cruz.
What mutations did they find?
Patient LS23 had a substitution of G to C at codon 2103. Patient LS41 actually had two mutations in the same exon of MLH1. One allele had an in-frame codon deletion, resulting in the deletion of a lysine. The other allele had a deletion insertion of two bases, which resulted in a lysine to alanine mutation at codon 618. Patient LS52 had a duplication in codon 213 of exon 3 in MSH2, which resulted in a frame shift mutation.
What did the researchers make of this information?
Sure, so let's go patient by patient. In patient LS23, the substitution in codon 2103 of MLH1 is interesting because it actually occurred in a splice site known to have high evolutionary conservation. This means that this variation was classified as likely pathogenic, or in other words, likely related to cancer risk.
Why is it likely pathogenic and not definitely pathogenic? From what I know, if a mutation messes with a highly conserved evolutionary site, that's definitely deleterious.
That's a great point. The thing is, mutations in splice site sequences could promote exon skipping, intron retention, or activation of cryptic splice sites. Not enough research has been done on these variations, so it's unclear exactly what the mutational effects on the MLH1 protein are in this particular case. Moving on to patient LS41, who had two mutations on the same exon of MLH1. The deletion of a lysine at codon 1853 is reported as having a pathogenic effect. Typically, it is associated with reduced MLH1 expression. Since many reports confirm the pathogenic effects of this particular mutation, the researchers considered this mutation to be the variant responsible for Lynch syndrome in that family. On the other hand, the lysine to alanine mutation in codon 618 was classified as non-pathogenic. It is not located in the interaction domain of the MLH1 protein, and therefore is likely not related to cancer risk. However, in a separate report, researchers urged to classify this mutation as a variant of uncertain significance.
What is a variant of uncertain significance?
Excellent question. The thing about genetic test results is that it's not often a positive or negative result like we're used to when we're testing for, say, strep throat. Sometimes we get variations in the genetic code that we simply just have never seen before, and so we can't know for sure if they do or do not contribute to cancer risk. These mutations are thus called variants of uncertain significance. Over time, and with the testing of more and more people, we will be able to make a more educated guess as to its role in cancer development. The last individual, patient LS52, had a duplication of a single nucleotide which resulted in a frame shift mutation in the MSH2 gene. The outcome of this mutation is the generation of a protein that is only 230 amino acids long versus the 934 that it's supposed to be. When cross-referenced across international databases, this mutation was not found, meaning this is the first report of this particular mutation.
So what now? What do these patients do with this information? What's the implication of this study?
That ultimately is up to the patient. With any genetic diagnosis or suspicion of hereditary disease, a consultation with a genetic counselor is recommended. These healthcare professionals help individuals and their families navigate through the medical, emotional, and familial implications of genetic disease. Patients can use this information in two ways. For themselves, their physician can use this information for preventative screenings or new medicinal and lifestyle changes. Or, patients can use this information for their family members and clinical issues that may be relevant to them as a result of their diagnosis. It can also play a role in family planning. In the research world, this study was important because it was the first conducted in a cohort that is not primarily Caucasian or Jewish. Different populations have different cancer risks, and so it's important to know these differences before we generalize our findings in the genetics field.
That is so cool. I want to get genetically tested. Where do I spit?